Hello, this is Todd Schlenke. I'm going to be talking about behavioral defenses that hosts use against parasitoids, and specifically the genetic and neuronal mechanisms that underlie those behavioral defenses. So, what are parasitoids? They're generally insects that infect other insects, although lots of caveats to that. And these are a bunch of images of different parasitoids infecting their hosts. Parasitoids are super diverse. I mean, pretty much for every insect that exists out in nature, there's some parasitoid or multiple parasitoids that infect it. Because they're so good at infecting their host, they often act as keystone species in natural ecosystems. And that heavy selection pressure that they put on their hosts means that hosts evolve defenses against the parasitoids, including immune defenses and also behavioral defenses. So how does the parasitoid life cycle work? Parasitoids are interesting because they're kind of in between predators and parasites. Like predators, they tend to kill their hosts, but they tend to live in or on their hosts for a long period of time, like parasites. Here's an example of a particular parasitoid wasp that infects fruit flies, Drosophila melanogaster. You can see that the wasp lays its egg in a fly larva, and the egg hatches, so you have a wasp larva in the fly larva. That wasp larva allows the fly to continue eating and growing, and it just consumes the hemolymph. But when the fly larva pupates, at that point the wasp starts eating the hard tissue and ends up killing the fly, and out of a fly pupal case, he closes a full-grown wasp. It turns out we actually know quite a bit about Drosophila behavioral defenses against parasitoids. For example, the fly larvae have a number of defenses. They avoid parasitoids by odor. They know what wasps smell like, and they avoid food sources that smell like wasps. If they do get poked by a wasp ovipositor, they'll initiate a rolling behavior, kind of like bucking bronco, where they try to dislodge the wasp ovipositor from their body. And fly larvae also medicate. They'll consume toxins in their environment to try to help them kill the parasitoids that are living in their bodies. There are also adult defenses. Now, fly adults are not infected by wasps, but they do change where they and how they lay their eggs to avoid their offspring getting infected. For example, they'll just reduce their oviposition when wasps are around. They'll avoid oviposition sites that smell like wasps. And they'll lay their eggs in more toxic places as a means of preventing infection of their babies. Now, one of the cool things about using Drosophila as a model for anti-parasitoid behavioral defenses is that we can connect the behavioral ecology and the organismal biology with underlying mechanisms like the neurobiology and neurogenetics. Okay, so I'm going to briefly explain some of the tools available when using Drosophila as your organism of choice. Obviously, it has a sequenced and well-annotated genome. For every gene in the Drosophila genome, there are mutant strains available. And there are genetic tools that are really useful, like the GAL4 UAS system. So if we look at the image on the right, basically the GAL4 UAS system allows you to express any construct you want in any tissue or at any time in the fly. And briefly, how it works is you have this GAL4 transcription factor. You put a promoter upstream of it, so it might be a tissue-specific promoter or a developmental stage-specific promoter that causes GAL4 to be expressed in a particular pattern. You also have a second fly strain, a responder UAS strain, where UAS is a promoter that attracts the GAL4 transcription factor, causing expression of whatever gene you want downstream of UAS. So if you mate a GAL4 driver line to a UAS responder line, you end up with a gene being expressed, whatever gene you want, in a tissue-specific manner or a stage-specific manner. And that allows you to overexpress any gene in the genome, knock down any gene in the genome. It's a really versatile tool. We also have in Drosophila a number of tools available for manipulating fly brains. For example, we have very specific GAL4 drivers that only drive expression in particular neuronal groups in the brain. We have transsynaptic labeling, where you can express a label in a particular group of neurons that you know is involved in your behavior to see what downstream neurons are also involved. The label moves from the upstream neurons to some downstream neurons, and that allows you to build a circuit of neurons involved in a behavior. And recently, we've also had the connectome. The connectome is basically where researchers used microscopy to identify and track every neuron in a single fly's brain. And so if you know a particular neuron is involved in a behavior, you can see, via these microscopy 3D renderings, every neuron that one neuron is connected to. And that's another way to build circuits in the fly brain and basically map out all the neurons that are in a circuit that define a particular behavior. Okay, so what have researchers found out about the mechanisms underlying fly anti-parasitoid behaviors? What I'm showing you here are cartoons of the adult fly brain, and the colored regions show regions that we know are involved in particular anti-parasitoid behaviors. So for example, the visual system, we know that flies are able to see wasps, and they know what wasps look like, because if you knock out photoreceptors in the retina, shown in red, the flies don't respond appropriately to the wasps anymore. We also know that once flies see wasps, the P1 neurons in the central complex of the fly brain produce less neuropeptide F. And having less neuropeptide F in the fan-shaped body, in purple there, makes the flies undergo behaviors like laying fewer eggs when wasps are around, or laying their eggs in more toxic places when wasps are around. So that's the visual system, and obviously there are a lot of parts of the circuits that we don't know about yet. All we know about are the photoreceptors and the neuropeptide F producing regions of the brain. Okay, we also know that flies can smell parasitoids. They know what wasps smell like. And they detect wasp odors via these two odorant receptors, OR49A and OR85F at the bottom of the image. Those project into the antennal lobes in light blue, which then project neurons into the mushroom body and into the lateral horn that seem to be required for fly behavioral responses. And once again, we don't know all the neurons in the circuit yet, and we don't know how the olfactory system interacts with the visual system, but we do know that all of these things are required. And in my lab, we're trying to fill in these circuits and understand every set of neurons that's involved. So here are some new unpublished data that we have, further filling in the fly visual circuit. You have a wasp image that the fly sees. It activates those pink photoreceptor neurons. And then we've shown that the L1, L2 motion sensing neurons are also required. And further downstream in the lobula, the LC11 neurons, which are responsive to small, randomly moving objects. Those are also required. So it appears that flies know what parasitoid wasps' movement looks like and respond to a particular kind of movement that they see in their environment. And understanding how these neuronal mechanisms underlying anti-parasitoid behaviors work, I think can inform us into the behavioral ecology of fly-parasitoid interactions. And I'll just leave it there and be happy to talk about that later. And finally, I'd like to acknowledge my department, the Department of Entomology at the University of Arizona, the NSF for funding, and especially my postdoc, Sean Davis, who's just an awesome Drosophila neurogeneticist who's done a lot of the work on fly behaviors recently in the lab. Thanks.